chromosomes are going to separate correctly, but the purple ones are going to do something bad, and you're going to end up with gametes with either an extra chromosome on the top or a gamete on the bottom, which is missing a chromosome. And so this is an assay we do cytologically to detect errors. But as geneticists, we actually have an assay which is using flies, and this is how we started doing things. So if we had a fly that we thought was a meiotic mutant, so let's start out in the top left here with a female who's got white eyes. Now the gene for white is on the X chromosome, so she's homozygous for a white mutation that gives her the white eyes, and we cross it to a wild type male that has red eyes. And if you do this and meiosis is working correctly, all females get one X from mom and one X from dad, and because the white mutants are recessive, you end up with a red-eyed fly. You also get white-eyed males for much of the same reason. So this is the phenotype when meiosis is working correctly. But if meiosis makes mistakes, then you get eggs that get either two X chromosomes from mom or no X chromosomes from mom. And if you get two X chromosomes from mom, like on the bottom left, you get a Y chromosome from dad. Now, flies are a little different than humans. XXYs are females. And if you have only one X chromosome, you're male. But what you can see here is the easily identifiable genetic phenotype. On the top, if meiosis is going well, you get red-eyed females. If meiosis is making mistakes, you get white-eyed females because they got the two X chromosomes from mom. And Bridges discovered this like 100 years ago and in flies. And so the idea that meiosis makes mistakes is something that's easily identifiable by genetic crosses. The one requirement is that the fly at the beginning where the meiotic errors are happening is viable. Now, meiosis happens in an adult, and there are many cases where there are genes required for meiosis that are also required for the fly to live. So we can't always do things this way. But I started my lab doing genetics this way, and we discovered a gene called sabido. And sabido, in flies lacking sabido, they were alive, but they made a lot of mistakes. We cloned the gene, and we found out that it localized to the spindle. And this sort of started us in a whole trek of things. So you can see that the sabido protein is localizing to that central region I talked about where the antiparallel overlap of microtubules is. Now, sabido made a lot of mistakes in meiosis, and there's a lot of reasons you can make mistakes in meiosis, and the cytology helps us figure out exactly what's wrong. And so, first of all, sabido co-localizes with many other proteins in the center of the spindle, including one called insem. And in the absence of sabido, these other proteins fail to localize correctly. And so what you're seeing here is in a sabido mutant on the bottom, there's no sabido protein on the spindle, and there's a couple things obviously wrong. First of all, sabido normally co-localizes with other proteins in the center. Those proteins are spread out all over the place. But more disturbing is that the spindle structure is disrupted. Here you're getting a tripolar spindle, and you can imagine if the chromosomes try to segregate, they're going to have a whole bunch of decision problems, and a lot of errors are made because there's, instead of two directions to go in, there's three. The other thing that's the real problem with sabido mutants is that central region of microtubules is missing, and you can see that on the far right here. And so the other proteins that normally localize there are missing, are not localized correctly. So sabido is a kinesin protein, and it organizes the spindle. It organizes that central region and allows other proteins to localize there. And so that got us into the meiosis business. But we realized that a lot of those proteins that were localizing in the middle, such as this insem protein, which is again shown here, these proteins are also required in mitosis. And when we knocked them out, we killed the fly. So we had to modify our approaches a little bit in order to study these other genes. And what I'm going to talk to you mostly today is what this protein insem is doing in the center of the spindle. So insem is part of the chromosome passenger complex. It's a complex that has four proteins. Insem is thought of as a scaffold, and it recruits two kinds of things. It recruits survivin and borealin, which are considered targeting subunits. This targets insem to certain structures. And as I'll tell you today, it targets insem to the chromosomes. Insem then brings in a kinase, and the kinase, of course, phosphorylates a lot of things. And in our case, you can think of the kinase as phosphorylating many proteins that allow spindle assembly to occur. So the CPC is thought of as a master regulator of cell division. But it's even more important in meiosis, as I'm about to show you. Okay, so as I showed you already, the CPC local, when we look at wild type oocytes, the chromosome passenger complex is located in that central region of the spindle. It's important to note here that the red, which is the incent protein, is on the microtubules. It's not on the chromosomes. And it's on those fibers that are going from pole to pole. When we treat the oocytes with colchicine, the microtubules are removed, and the chromosome passenger complex, or incent, shown here in the bottom, actually moves onto the chromosomes. So incent and the CPC can bind the chromosomes, but only when the microtubules are absent. So in short summary, the chromosome passenger complex can be in one of two places. Normally we see it on the microtubules, but if they're absent, you'll see it on the chromosomes. So how does that relate to the function of incent, or why is this important to the spindle? So how would we address this? Now, I already mentioned that if you make a mutation in incent, the fly dies. So what's been really advantageous is using a very powerful tool in Drosophila genetics, which is to do tissue-specific RNAi. So again, if you knock out incent or any member of that 4-protein complex, the fly dies. So what we want to do is knock out the gene only in the oocyte. So we do that by using RNAi. Now, the shRNA shown here on the right has a promoter for what we call UAS. And that is a yeast promoter, and so normally it's not expressed. But if we make a fly that also has the GAL4 protein under a tissue-specific enhancer, now the shRNA molecule will be made in the tissue specified by the GAL4 driver, as we call it. Now, so in one set of experiments, we combine the tissue-specific enhancer with the shRNA to knock out the gene in a specific tissue. We're going to use something called MAT-alpha GAL4, which expresses GAL4 only in the oocytes and therefore depletes genes in an adult fly, but only in the ovary. And we'll look at what happens when we deplete genes using that method. Then what we're going to do a lot of is add a third thing, which is an RNAi-resistant transgene. So all three of these things are in the same fly. The RNAi is knocking out the endogenous copy of the gene. And then at the same time, we're expressing a transgene, which has been made resistant to the RNAi by a few silent changes. And then we'll mutate this molecule to address function. Okay, so that's the basic method that we're using for this talk, is knocking out the endogenous gene, seeing what happens, and then bringing back an RNAi-resistant form, often mutated in a certain way. So when we did that, when we knocked out incent, we got a relatively surprising result. Remember, when we knocked out Sabido, we got an abnormal spindle. When we knock out incent, we lose the spindle. We get that spindle null phenotype. So this told us that the chromosome passenger complex is required to assemble all aspects of the meiotic spindle. So we're talking about the microtubules of all those two kinds of microtubules. It's required, SPC155R is a kinetochore protein, and it's also absent. So it's also required to build the kinetochore, which is one of the main structures for binding to microtubules. All right. So, and this is using a drug to do the same thing. BN2 is a drug that inhibits the kinase, Aurora B. And so you can see that in the absence, when we treat the oocytes with that drug, the microtubules disappear. Also, the Aurora B kinase can be shown to be eliminated by absence of targets like phosphorylation of incent. Later on, I'll talk about the antagonism of all of this, which is WDB is a PP2A, and so phosphatases are also involved. And so if you knock out the phosphatase, you can actually save the spindle. But I'll talk about that a little later. But the key point here is that if you inhibit remove the chromosome passenger complex or inhibit the kinase, you get no spindle. So we were interested in how does the chromosome passenger complex promote spindle assembly? Because this is the question we're trying to answer. Remember, the chromosomes in oocytes are the focus of spindle assembly, but the mechanism of that is not known. And this phenotype, seeing the complete absence of the spindle, made us think that studying the chromosome passenger complex would allow us to understand how the chromosomes direct assembly of the meiotic spindle. Okay, so how does the CPC promote spindle assembly? That's the big question we want to address. So again, it's the chromosome passenger complex involves these 4 proteins, with 2 of them being targeting subunits. We started out with a wild-type transgene that was RNAi resistant, and that could rescue the RNAi. And so, and then what we did is to study basically how does the chromosome pass-through complex get recruited to chromosomes. So we started to mess around with mutants on targeting of the chromosome pass-through complex. I already showed you that the chromosome I'll call it CPC because I'll keep stumbling, but I already showed you that CPC can be seen in two places, the chromosomes or the microtubules. So where does it really need to be to organize a spindle? So we generated a whole bunch of mutants manipulating its localization pattern to ask the question of where does the chromosome pass-through complex have to be in order to build a spindle? So we made a whole bunch, this is just to show you that we made a whole bunch of RNAi-resistant mutants and tested their function in oocytes. And I'm going to go over a couple of them that are the most important. So the first thing we did was target the chromosome pass-through complex to the centromere. What would happen if we forced, remember, the idea was that we see the chromosome pass-through complex in two places, let's force it to one of those places and see what happens. So we force it to the centromere. And when we did that, what you're seeing here in wild type normal bipolar spindle, and we did it by either fuse, and what we generally would do is remove the targeting domain. The N-terminus region is where those targeting subunits bind. We'd remove them and replace it with a protein that could target it. So MIS-12 is a kinetochore protein, and Sabido or Fio are microtubule binding proteins. So we're asking, what happens if we target the CPC to either of these structures? And what you can see in these pictures is that if we target INSEMP to the centromeres or kinetochores, we get kinetochores, we get microtubules growing from the kinetochores, but we don't get the full bipolar spindle. We don't get the central spindle. That's what's really missing here. We're getting wonderful kinetochore assembly. On the bottom here, you can see in white, SPC105R kinetochore protein assembling quite nicely and building microtubules, but we're not getting a bipolar spindles, and we're not getting those microtubules in the center, the central spindle. So we're getting half a spindle. We're getting a spindle where we targeted the microtubules, the CPC. And you can sort of, we quantitate all of this stuff. So on the left, you're seeing the wild type with lots of SPC105R foci per oocyte. You actually expect 8 because there are 8 centromeres at metaphase 1. And when we target MIS-12 INSEMP to the centromeres, you get lots of kinetochores too, but you don't get a full spindle. So what happens if you target CPC to the microtubules instead? So we did that with by fusing and I should point out in this picture that the key part here is what we call the inbox, because the inbox is the part of INSEMP that recruits Aurora beam. So these little substrates on the bottom directing MIS-12 directly fused to the inbox. It forces the inbox to go to the kinetochores and will bring in Aurora beam. And so if instead we fuse inbox to Sabido, so we're trying to force it to the microtubules, we don't get anything happening. So again, if we force the CPC to the centromeres, we get kinetochores and we get microtubules. If we force the CPC to the microtubules, we actually don't get any spindle assembly at all, which is kind of surprising. So it looks like you at least need to, so then we thought, well, we've got two targetings. We're independently targeting the CPC to the two structures, the microtubules and the kinetochores. What happens if we do that at the same time? So we co-expressed the targeted inbox to MIS-12 and also the sub, and we do that in the same oocyte, and it didn't help things very much. We got a little more spindle assembly, but we really only got kinetochore fibers. You can see there's no bipolar spindle here. So, like in these top two pictures in these controls over here, this is the bipolar spindle in the wild type. You're obviously not getting that, even though we're targeting the CPC to the two structures that we normally see it at. So this made us think that in order for the CPC to work, it has to be able to do both things at the same time. That the CPC has to, because each of these constructs here can only do one of the two things. We thought maybe together they would cooperate, but they don't. So instead, that INSEMP intrinsically has to be able to do both things at the same time. Now, to help us understand this, we took advantage of another observation. And that observation was that when we knock out the kinase, so we knock out Aurora B, you see something kind of interesting. That the rest of the subunits still localized to the chromosomes. So if we knock out INSEM, we get no spindle, but other subunits of the CPC, such as borealin, targeting subunit, it also doesn't localize. And then there's a protein called HP1, which I want to introduce now, which is a chromatin protein. And it is found on the oocytes, chromosomes. It's enriched in heterochromatin. So it's a heterochromatin protein. And we noticed that, almost through serendipity, that when we knock out the kinase activity, the other three parts of the CPC localize to the chromosomes. So borealin is shown here, localizing to the chromosomes, and it co-localized with HP1. This was really striking. Because HP1 is a heterochromatin protein. It's not on all of the chromatin. It's only on about half or a third of it. So you can see that the CPC and HP1 are co-localized, suggesting that the CPC is first able to be recruited to the heterochromatin region of the chromosomes. And you can see that with the other subunits, survivin and INSEM, all of them co-localizing with HP1. So this made us think that we should be looking at HP1 as interacting with the chromatin. So the other thing that we did is, knowing that borealin and survivin are required, was to test them individually. And so we took our INSEM transgene and took off the targeting subunit in white here and replaced it with either survivin and borealin. So now we're testing what is survivin capable of doing, or what is borealin capable of doing. And what we found is that borealin was very good at rescuing the spindle, but survivin or borealin was not. And I apologize for Drosophila nomenclature, but in Drosophila, borealin is called survivin. Okay, so this survivin subunit of the target in red here, the targeting subunit, is also called borealin. Okay, so I apologize for a little bit of the intermixing of the terms there. Survivin and deterrin. I'm sorry. So survivin is also known as deterrin in flies. So you'll see me call it that. Borealin is a separate subunit. I just mixed up my terms there. See how hard this is to do. So when we fused survivin to INSEM, we got partial spindle assembly, meaning the kinetochore is formed again. But what you can see here is that the INSEM, which is now fused to survivin, is really sticking to the chromosomes. But borealin, when fused to INSEM, almost got full rescue. You can see it here with a bipolar spindle. In red, you can see INSEM, which is here fused to borealin in the central spindle. So borealin seemed to be able to do everything INSEM had, to target INSEM for everything it had to do. It can go to the chromosomes. It can also go to the spindle. Survivin, however, was only capable of recruiting INSEM to the chromosomes and directing kinetochore-based things like kinetochore microtubules. So borealin seemed to be sufficient to target INSEM for spindle assembly. And if we go back to the previous thing I just said, where I was talking about how we need both chromosome and microtubule targeting, it seemed that borealin could do both things. It could target the INSEM to the chromosomes, and then it could target it to the microtubules. Survivin was stuck on the chromosomes. So looking at borealin became very important. It seemed to be able to do the task that INSEM would target INSEM correctly. And it could even do it without the other subunit. So when we fused borealin to INSEM and we removed the other subunit, deterrin or survivin by RNAi, you still got a bipolar spindle shown on the bottom right. So borealin was sufficient, and it seemed that the only role of survivin is to help borealin be recruited to the CPC. But once you fuse borealin to INSEM, you don't need really survivin anymore, except to help maybe stabilize the spindle. So we looked carefully at borealin, and one of the things that other researchers have known is that borealin has an HP1 binding site. So we got very excited about this because I just showed you that the CPC co-localizes with AP1 on the chromosomes. So we came up with a hypothesis that borealin brings the whole CPC to HP1 on the chromosomes. Now to test this, there's a competing models for how borealin and survivin target the CPC. And that is that borealin and survivin are already known to interact with other chromatin signals. Survivin interacts with a histone modification that is directed by something called haspid kinase. And borealin interacts with an H2A modification that depends on another thing called BUB1 kinase. And in fact, most of the literature in terms of targeting the CPC to the chromosomes focuses on the roles of haspin and BUB1 kinase. The role of HP1 in all in targeting CPC, there's only a small number of papers on that. The majority of the literature on targeting the CPC of the chromosomes involves BUB1 and haspin kinase, targeting borealan and survivin. So we were sort of suggesting something new, and we sort of knew that we were in the right direction because we knocked out haspin and BUB1, and we even knocked out both of them by RNAi, and it didn't seem to bother spindle assembly. So spindle assembly was normal. This is our assay. I talked about our genetic assay. Fertility and chromosome segregation assay genetically were normal. Non-disjunction was very low. So these pathways that have been proposed as so important for localizing the CPC to the chromosomes didn't seem to be too important in meiosis of oocytes. But we thought HP1 was. And furthermore, as I already mentioned, INSEMP or the CPC co-localizes with HP1 in the absence of the kinase activity. That depends on INSEMP. It also depends on the other subunits. And that HP1 itself localizes to the chromosomes in the absence of the kinase. But we also noticed that HP1 was on the microtubules in a wild-type spindle. And this is shown again on the top. Here's a wild-type spindle with HP. Now, so HP1 is kind of not as tight as the chromosome passenger complex, but it does co-localize with the microtubules rather than the chromosomes in a wild-type spindle. And importantly, if we inhibit Aurora B, so there's no kinase, INSEMP stays on the chromosomes, as does HP1. So this led us to a model that what the kinase does is allow HP1, that step one of spindle assembly was the chromosomes recruiting the chromosome passenger complex. And step two, go back a step. Not only the chromosomes recruiting the chromosome passenger complex, but HP1 recruiting the chromosome passenger complex to the chromosomes. And then the kinase would enable both of these, INSEMP and HP1, to move on to the microtubules. So we tested that in a couple different ways. The first way was to mutate borealan in its HP1 binding site. And when we did, so the HP1 binding site, as I showed you in the previous picture, is in the C-terminus of borealan, which again, remember, is fused to INSEMP. And when we did that, spindle assembly didn't occur. And that's shown on this picture over here. Borealan delta C is the mutant that cannot bind HP1, and in black here is the frequency of absent spindle. Also, pointing out here, borealan delta C, the number of kinetochores formed is also much reduced compared to either wild type or the borealan INSEMP fusion. And so just notice that here's the wild type spindle, here's the borealan INSEMP fusion, indistinguishable in terms of kinetochore formation, but if you remove the ability of borealan to interact with HP1, no kinetochore assembly. Okay, so this led us to think that the HP1, which is recruited to the chromosomes by a histone methylation in heterochromatin, recruits borealan, and that brings in the rest of the chromosome passenger complex. Then Aurora B is recruited by the inbox. Now it's known to phosphorylate a bunch of stuff, including H3S10, which notices the amino acid next to H3K9, and it's known that H3S10 phosphorylation can actually disrupt the interaction between H3K9 and HP1. So this led us to a model of how the chromosome passenger complex moves from the chromosomes to the microtubules. That Aurora B phosphorylation triggers a dissociation of HP1 from the methylated histone, and then it can move on to the microtubules. So to test this is the next two slides, and these are dense slides and are the most complicated part of my talk. So I'll go through it a little bit slowly. But what we're trying to do is test this model and test if Aurora B kinase actually controls whether or not HP1 and the CPC associated with the chromosomes or not. So we can do that by inhibiting Aurora B. So on the far left, we have a wild-type spindle, and here's CPC subunit deterrent on the spindle, and HP1 also on the spindle. If we inhibit Aurora B kinase, and the neat thing about these experiments is the drug is applied after the spindle assembles. So you can actually think that when the drug is applied, the spindle looks like the picture on the left. And then we apply BN2 that inhibits Aurora B, apparently phosphatases take over the shell and the spindle disassembles. Now, when the spindle disassembles, what happens? The turn moves from the microtubules back to the chromosomes. HP1 moves from the microtubules back to the chromosomes. And so these experiments show that the CPC components like INSEMP and deterrn and survive and borealin can move from the microtubules to the chromosomes and back, including HP1. These are experiments using that methyl mark. We can just skip over those. And this experiment I'm pointing to here is the borealin INSEMP fusion. It does the same thing. We treat that with BN2. The INSEMP jumps off the microtubules, goes back to the chromosomes, and so does HP1. And notice that INSEMP and HP1 are co-localizing. And also notice that when we delete that part of borealin and interacts with HP1, INSEMP no longer goes to the chromosomes, even though HP1 is still there. So this supports a hypothesis that that the CPC interacts with HP1 on the chromosomes, and because when we delete that region it no longer does, it supports the idea that borealin is interacting with HP1. Okay? But that the Aurora B kinase is actually the thing that's causing these components, the CPC and HP1, to move from the microtubules to the from the chromosomes to the microtubules. So to test if it's really the kinase, is we use colchicine. So again, we're treating oocytes that kind of look like this with a drug that gets rid of microtubules. Now BN2 gets rid of microtubules because Aurora B is needed for microtubules. Why colchicine gets rid of microtubules, I don't actually know. But I know that it doesn't inhibit Aurora B. And so now we're testing what happens if the microtubules are gone, but Aurora B is still active. And so what you see, as I showed you before at the beginning of the talk, is that the chromosome processor complex goes back to the chromosomes. Shown here with deterrn going back to the chromosomes. But notice how faint HP1 is. And let's just focus on these last two. So in this panel, we're using an INSEMP mutant, which can still bind to microtubules. Okay? And the one on the right is the delta C mutant that cannot bind, that borealin is defective in binding HP1. Okay? So even though it says INSEMP delta HP1, that is deleting part of INSEMP and not borealin. So this INSEMP genotype can bind HP1 and assemble spindles, and this one cannot. And what you're observing after colchicine is that INSEMP moves to the chromosomes, but HP1 does not. Similar over here is wild type, that with colchicine treatment, the CPC goes to the chromosomes, but very little HP1 does. And just compare these two at the end, because this version of borealin INSEMP cannot bind to the chromosomes, and therefore cannot recruit Aurora B to the chromosomes. Note that HP1 is very strong on the chromosomes. So these experiments show that if Aurora B is on the chromosomes and active, HP1 will not be. Okay? If Aurora B is inactive, HP1 can be on the chromosomes. So if you combine these two experiments, you end up with this kind of a model. And in this model, a trimeric complex, INSEMP and those two targeting subunits, borealin and survivin, are recruited to the chromosomes by these little gray boxes, HP1. At that point, they recruit Aurora B. Aurora B is the little orange guy. Now, the Aurora B is the kinase that phosphorylates a lot of stuff. It phosphorylates probably kinesins that help the spindle assemble. It probably phosphorylates SBC105R and other kinetochore proteins so you can build a kinetochore. But it also phosphorylates H3S10 and other chromatin targets. And that forces HP1 to leave the chromosomes. That's what these two experiments show, that Aurora B forces HP1 to leave the chromosomes. It also forces the CPC to leave the chromosomes, maybe because it's interacting with HP1, and the CPC, because of things like borealin, also binds. And this is why borealin is so important. Because borealin can bind to HP1 and it can bind microtubules. And so now the CPC moves to the microtubules and it's focused into the center by that kinesin subito that I referred to at the beginning. Okay? So this is very satisfying for us because it explains chromosome-mediated spindle assembly. Because the important thing is that this happens in a very big oocyte, but you only want spindles to form around the chromosomes. So it focuses things on the chromosomes by localizing Aurora B to the chromatin. Then, once Aurora B is active, it can move to the microtubules that are close to the chromosomes and have the rest of the things happen. Okay, so by having HP1 being a key activator, it forces the CPC to be active only near the chromosomes. And that's what so the chromosomes recruit the CPC, it is then ejected, and it accumulates on the microtubules and gets, and its accumulation in the microtubules is modified by kinesis. Okay, so that's a satisfying idea about how we build a spindle. But it doesn't tell us why do you really need the chromosome passenger complex on the spindle. It's pretty obvious why it has to be on the chromosomes. It has to recruit the microtubule assembly factors to the chromosomes. But why does it have to move onto the spindle? Why does it have to be at this central spindle? And so that's a clue to that came up in some of our analysis. Notice that when we for example, when we fused survivin or borealin, we got only full spindle assembly with borealin, and survivin was partial spindle assembly. So why is there this difference? Why do we need to have a central spindle with the CPC on it? And a clue to that happened when we were messing around with our transgenes, and we were testing now in a wild type background. So if we have wild type INSEM present, and we also express either Mis12 inbox or sub-inbox, what happens with chromosome segregation? So what we're doing in this case is we're building a normal spindle because the wild type INSEM is there, but we're also forcing extra Aurora B kinase, either to the kinetochores with Mis12 inbox or to the central spindle with sub-inbox. Okay, because sub targets the microtubules, Mis12 targets the kinetochores. The literature said that if you target the Aurora B, if you overexpress Aurora B at the kinetochores, Aurora B will cause the kinetochores to make some mistakes. But we observed the opposite. When we forced the CPC to the kinetochores, as shown in this graph, the error rate was low. Biorientation is a good thing. So wild type does it mostly about 100% of the time. When we forced the overexpression of Aurora B at the kinetochores, there were very few errors. But when we forced the chromosome passenger complex to the central spindle, there were a large number of errors. It looked like the active place, that the central spindle was the place where Aurora B was to influence biorientation. It was on the chromosomes to build the spindle, but it would move to the microtubules in order to regulate the segregation and biorientation process. And we had other mutants that built other mutants in the chromosome passenger complex, like INSEM mutants that could build a spindle, but they still made lots of errors. And so by looking at all of these mutants, we came to the hypothesis that the chromosome passenger complex had to be on the microtubules in order to regulate the biorientation process. What we're talking about is this picture again. How do these errors get corrected? Most of the literature would suggest that Aurora B at the kinetochore is the thing that corrects the errors. That when Aurora B is present at the kinetochores, it can sense these mistakes. And it can cause the microtubules to, the erroneous attachments to detach, as shown here, and reattach and do the correct thing. So the model was CPC at the kinetochores did error correction. However, we began to believe that it had to be on the spindle. And there are a couple ways that we're thinking about that. So the rest of this talk is kind of speculation with a little bit of data. And there's two important pieces of data that are guiding our thinking on this problem. How does the problem being, how does the chromosome passenger complex on the spindle regulate what the chromosomes are doing? The first has to do with PP2A. That when we knock out PP2A, in a separate project, we knocked out the phosphatase. This phosphatase antagonizes Aurora B. Aurora B phosphorylates things, PP2A dephosphorylates those things. When we knocked out Aurora B, we didn't get those microtubule attachments, those end-on microtubule attachments that allow the chromosomes to move to the poles. What you're seeing here are lateral attachments. That the microtubules are not ending at the kinetochores. The kinetochores seem to be moving along the sides of the microtubules, and that's graphically assessed over here. In wild type, most kinetochores are making end-on attachments, like shown in this picture, but in PP2A knockouts, most kinetochores are making lateral attachments, like shown in this picture down below. So PP2A activity, removing the phosphorylation of Aurora B, seems to make kinetochores detach from the microtubules. The second observation was what the kinetochores themselves are doing. And it's contrasting two kinetochore proteins, SBC105R and NDC. When we knock those two guys out, what we see is a kinetochore null phenotype I showed you at the beginning. Instead of at the bottom left here, so now we're knocking out the kinetochore proteins. When you knock out kinetochore proteins, instead of the kinetochores nicely pointing towards one pole or the other, they're sort of scrambled all over the place. SBC105R knockout or NDC8, two kinetochore proteins we knocked out. They're kind of scrambled all over the place. But there's a big difference between them. When we knock out SBC105R, the kinetochores are randomly associating relative to the microtubules. Some of them seem to be touching the microtubules, but others are not touching any microtubules at all. Again, in contrast to the wild type situation where kinetochores are making end-on microtubule attachments. NDC80, and so again shown here, and it actually measured. We measured the distance from the kinetochores to the microtubules, and you can see in many cases the distance from the kinetochore to the microtubules was quite large. When we knocked out NDC80, also killing what we thought was killing the kinetochore, the microtubules were still close to the kinetochores. You can actually see that in this picture. The kinetochores, although they're kind of scrambled all over the place, they seem to be all associating or riding along the sides of microtubules. So the conclusion was that if we knocked out kinetochore protein SBC105R, we lost all connections to the microtubules. But if we knocked out NDC80, we retained those lateral attachments. So SBC105R, which is present in the absence of NDC80, must be capable of making lateral attachments. Okay, so that led us to this model, which is that the way that bioorientation works is for the chromosomes to associate laterally first, and they figure out their bioorientation, and then they make end-on attachments, and when they've done it successfully. And so, so the hypothesis kinetochores to SBC105R first establish lateral attachments, error correction is done, and then they transition to end-on. So it's important to not make end-on attachments until all errors have been corrected. So where PP2A comes in is remember that PP2A is in the absence of PP2A, you get only lateral attachments. So that tells us that the dephosphorylation reaction promotes end-on attachments. So this transition from lateral to end-on depends on the phosphatase. Or, said another way, that when the kinetochores are phosphorylated, they do lateral attachments. And that's what this model is showing, that when kinetochores are doing lateral attachments, they are phosphorylated, and when they make end-on attachments, they are dephosphorylated. Now, if I combine that picture with what we know about the chromosome pasture complex, remember the chromosome pasture complex is located in the center of the spindle. And so when it's in the center of the spindle, you can imagine it's very close to the kinetochores, and Aurora B can be doing this phosphorylation event. But as the kinetochores start to make the right decision, they start moving towards the pole, and they move away from the chromosome pasture complex, so the phosphatase can take over and maybe dephosphorylate the kinase, and then you make end-on attachments. So our model is that the way bioorientation works is that initially the kinetochores are making lateral attachments, and they are phosphorylated. They begin to slide away from each other and towards the poles, and when they do that correctly, they separate far enough that they leave the area of CPC, the kinase becomes diluted, and the phosphatase removes the phosphate, and they make the end-on attachments. What's key here is that the kinase is spatially restricted to the center of the spindle, and this is perhaps why the microtubules, the CPC on the microtubules is so important that it has to be restricted on its location. It's restricted to the central spindle, and it creates an environment where you make lateral attachments. But as the kinetochores make the right decisions and move towards the poles, now they leave that area of high kinase activity, and the phosphatase takes over, and you make endon attachments. So this is the reason why we think why the spindle localized, and more precisely, the central spindle localized CPC has to is so important for regulating bioorientation. By spatially restricting the CPC, the kinetochores can literally move away from them if they're doing the right thing and become dephosphorylated and make endon attachments. So we're testing this model much in the same way that we did with INSECT. We use an RNAi-resistant SPC1FR transgene, and it's actually known from a colleague of ours named Tom Maresca that the N-terminus region of SPC1FR can actually make those lateral attachments. And the hypothesis is that by the phosphorylation, there's 2 phosphorylation sites for Aurora B located in this region, that controls this activity. So when this region is phosphorylated, we propose that SPC105R can make lateral attachments. When this region gets dephosphorylated, it converts to endon attachments. And so we're testing this with these mutants. And the preliminary evidence is that if you delete this region, you make lots of bioorientation defects. So we think we're on the right track. But we're right now making mutants to mimic phosphorylation or not phosphorylation to test the hypothesis that phosphorylation of this region regulates whether or not the kinetochore makes lateral attachments or endon attachments. There's actually some evidence out there that this is actually important. Trivedi et al. In the Stukenberg lab have actually, much of our thinking actually came from their lab because they proposed actually that CPC on the microtubules is actually the thing that phosphorylates kinetochores. And so they have a whole mathematical model, which I barely understand, that it describes how microtubule-associated CPC phosphorylates kinetochores. I really need to try to figure out this model more. And also Iva Tolek, who suggested that kinetochores ride along bridges of microtubules, which we refer to as the central spindle, and that they use this, what she calls a bridging fiber, and we would call the central spindle in order to separate. So there is some interesting evidence out there which is consistent with our suggestion that the central spindle is so important for regulating what the kinetochores are doing. And we are advancing more with our imaging to look at higher and higher resolution, and for this we are entering into the world of things like deconvolution, but also other forms of super resolution microscopy. So we can really look at with higher resolution how the kinetochores are interacting with microtubules. But even without some of that super resolution, you can see like really nice patterns. So here's a wild type spindle, and you can see pairs of kinetochores interacting with the same bundle of microtubules. Like if you can just look at this one here. And this is exactly what we think. This is Iva Tolek's bridge between the microtubules, and it's mediating the separation of them. And as they move away from each other along this bridge, they leave the area of, in this case shown in white, of chromosome passenger complex. So that's where we're going, trying to see how the CPC on the microtubules regulates the kinetochores. A lot of this work was done by a grad student named Lining Wang, who's since left the lab. So she's not, I have an undergrad who likes to make monthly themed photos of our lab. And so this is our April one, and this is the current lab. And it reminds me to point out that Janet did a lot of the work on with Lining. Lining did most of the work on CPC with Janet's help. And Janet did a lot of the work on the PP2A story, and Neha I always forget the names now. But Neha and a number of these people, including anyway, were Okay, so right. So again, the names of the people. Oh, good. Yes, I forgot. Jay here with these ears on. He's working on the XP75R project. And so again, the names of the people in the lab. A lot of so what's wonderful in working in the fly community is a lot of wonderful collaborators, and particularly Christian Lanner, who's given us a lot of reagents that have helped us do the work we're doing. I'd like to thank the organizers again for inviting me, and I'd love to take questions. Thanks.
Okay, thank you very much.